Okay. The music is not so critical. You would have heard this ongoing music in the brain. Any area, any neuron is producing this well-known phenomena of a spike. I'm going to talk to you about spikes today, trying to understand how to model them automatically. But before that, here is my plan for the talk. I want to start with some excitements that I think we should have felt more here, because I think some phenomenal things are happening now today in the world. We all should be aware about that because we are brain researchers. And I don't know if you know that we are really in peak times of excitement about the brain at the beginning of the 21st century. Then yesterday there were some issues about what does it mean to understand. Can we understand via simulations and not? I would like to discuss that a little bit if possible. Then a little bit of history about the spike and lessons that we can get from those who modeled spikes, Hodgkin Huxley. Then I want to speak a little bit about my work together with Henry and Kevin Martin and Bert Sackman, and then show you some automatic way, I think very successful one that we have developed recently to model these spike patterns in different neuron types. With some highlights of the present work. And if I will have time, I will speak about, I hope to be able to tell you a little bit about this, what I see today as a new brain agenda that we are part of, but I think we should say it explicitly, that we are now developing a new brain agenda. It's not a continuous of what happened before, and I think we should all be very excited and enthusiastic about what's happening. And if time will remain, but I guess I don't have music, so I prepared for you a movie about dancing neuron, but this neuron dances with music, and without music it doesn't know to dance. So, okay, let me start with an event that happened at the Hebrew University a year ago. There was an international committee, 2 Nobel laureates, Bert Sackman and Richard Axel, and other top scientists came to Israel to advise us about how to develop a new brain center. We were hoping to get a fantastic amount from a very rich lady, which we worked a lot to get it from her, and we did. So the question is how to build a new center, and they said that the following, which I think is very important. Great universities will be judged in the coming decades by what they contribute to our understanding of the human mind. Indeed, some believe that our survival as species depends on better understanding of human actions, perceptions, matters, patterns of thoughts and emotions. Fortunately, you at the Hebrew University have an excellent foundation on which to build. Computational and theoretical neuroscience should remain a central core of neuroscience community because of its excellence and because of its ability to link and unify different experimental events. They wrote a 100 pages suggestion about how to build a new center. Anybody of you who would like can look at it. I think it's a fantastic roadmap about where we are heading. They worked very hard. We really thank them. And I think what's happening now, as I said, is something like that. There are several centers in the world that are built in this kind of scheme, trying to connect quantitative analysis at the neuronal circuit level. And I think this is the level we are starting to focus in the next 10, 20 years at a very fine level, connecting it to disorders and cognitive functions. This is the structure of the Hebrew University Center with 15 new faculty positions, but it's happening all over the world, and that's the excitement. I don't think that I have never dreamt that there will be at the same time almost these and actually many more new centers. For example, I'm just coming from London, where I'm doing a short sabbatical. There is a very big excitement about Sainsbury and Wellcome contributing together something around $300 million to create a new center with 12 new faculty and computation and neuroscience core. So there is a core in all these centers of modeling. There is no new center in the world now without a modeling, a significant part of modeling, because of what was written before. We have Rodney Douglas and Kevin Martin in Zurich. There is this center in Paris. There is the Brain Mind Institute Henry Markham in Lausanne. There is the Janelia Farm. I have some reservations about Janelia Farm. We can discuss it later. A lot of money went there, and so forth and so on. $800 million for the new center in Colombia with Larry Abbott and others. Amazing times, really amazing times. Something is happening. We have to be aware of it. You, especially the young generation, will be part of it. We have to realize that something very important is happening in brain research, not in any other field, in brain research. And there is a very good reason to it, because I think we will understand the brain by the end of this century, and whoever doesn't feel so should not go to brain research. Those who feel so should go to brain research, and this is the time to go to brain research. Maybe we can talk about this later. I just want to highlight what's happening all over the world. And of course, in Japan and in Natal, Miguel Nicolelis is building a center, and I didn't mention all the rest. But it's an amazing time for me. I'm personally very excited. I want you to feel the same. Okay, but we do not understand the brain still. Why? Why don't we understand the brain? And why do we understand the car? We can say that we understand the car. Of course, we built the car. There was a purpose for building this car. There was also a purpose for building a brain. But in this case, we built the car. And in this case, evolution. You see, evolution built the car. So why do we understand the car and we don't understand the brain? We understand the car, first of all, because we have a blueprint of the car. We designed the car. We also know what each part of the car is doing, because we designed it. We don't know what each part of the brain is doing, but we have to study that. We also can simulate a car. So before we build the car, we can simulate the car in details. And this simulated car should do what the car should do before we build the car. So simulation is critical, and we'll decide this later about what Henry Markham's vision is, because I'm a strong believer in this direction, as you understood yesterday, and I will try to convince you later why, if I have time. Then we should model a car. I don't believe that simulation per se is understanding. We should model the car. And later, if I have time, I'll try to distinguish between simulation, modeling, and theory. These are not the same things, although there are some overlaps. Anyway, we can learn from the car, saying that we understood the car. We also understood the heart. We understood some parts of us. We did not understand the brain, but we can understand the brain because it is a machine. Complicated machine. It's a machine. It's a changing machine, but it's a machine. Some people, sometimes when I say that, especially when it's to public audience, they really come in riots because they don't like to feel machines. I like to feel machine, actually. A nice machine, but a machine. Okay, so what is the problem here, of course? The problem in brain is that we have all these levels. And this was taken from the book of Terry Chesnovsky. Long time ago, there are all these levels of description. Each one of us is focused very much on his or her level of description. We fall in love with synapses, or with circuits, or with the other circuits, or with cognition, but we don't know how to link them. So we get more and more information at a specific level, and there is no theory, there is no way that we know how to link them. And the Blue Brain Project is one way, not the only way, but one way to link levels. Typically, we know today to link a level to another level. Let's say Hodgkin-Huxley model linked, that's why we use in most of the books from neurons to something, from something to something. So Hodgkin-Huxley is from ion channels to spikes. So we know to do one from one level to the next. We don't know to do many froms, from molecules to behaviors. And we don't need necessarily to do all this, because when I'm talking now to you, I don't think I need to know the calcium concentration in my spine now. Later, I may need to know it because maybe I studied something and there is some plastic processes, but even that I can formulate simplified, and I don't need to know all the genes that are working now necessarily, depending on the question. We'll discuss it later. So this talk is about 3 or 4 hours talk. Okay, so because of all these things, and I will go deeper into each one of the issues, I really believe that we need this. We cannot understand the brain without having a theory about the brain eventually. But along the path between theory and the details, we will do some simulations, we will do some modeling, and eventually there will be a theory about the brain. Without a theory about the brain, I really don't think that I can say understood, really understood. I got some understanding along the way, but a deep understanding in my mind is really high-level theory. And I always like to cite William Thomson, or Lord Kelvin, a great mathematician, who said the following. I don't know if you can read it from there. I am never content until I have constructed a mathematical model of what I'm studying. If I succeed in making one, I understand. Otherwise, I do not. So I'm sure I can convince you easily this is a group of neuroinformatics that without modeling we don't understand much. And my mentor about all that I'm doing today is, of course, Will Roll that Eric mentioned. He was the one who really taught me how to think about neuroscience, about dendrites as well. And he was doing the following. So he knew about the point neuron, of course, but this point neuron, which was very popular in the beginning of the 20th century, actually the first half of the 20th century, all modeling was done with a point neuron. He realized, Will Roll, that something is wrong with this oversimplification for understanding signal processing in the brain. So he was starting to climb, so to speak, in terms of the complexity of models. He went up until this level, and then I will show you that he reduced. And this level for me is what Rodin did for the kiss. The kiss of Rodin in Paris is a simulation of a kiss. It's a beautiful sculpture. You cannot resist going to the Rodin Museum in Paris and see this kiss, but this kiss is a simulation of the kiss. And the blue brain is Rodin kiss, is a simulation. When you look at this sculpture, you feel that you understand something about kissing. And this is good, especially when we talk about kissing. But the thing is that you don't want to stay at that level when you model. And as Roll told me very well, you have to go backward into a simplified version of the kiss, of the neuron, of the brain, of the column, into a level that is sufficiently detailed to describe your phenomena, but not too detailed to replicate your phenomena, because this is not full understanding. So we are here, and of course artists were the first one to do it, and Brancusi kiss is a theory of the kiss. So this is simulation of the kiss. This is the theory of the kiss. If you reduce Brancusi a little bit more, you will become a point neuron, and it will not be a kiss anymore. Okay? So we are talking about what level of description is the correct level, and theory, of course, is the way to capture the essence. So we should learn from artists. That's why I went to learn from artists and had this. I'm very proud of the book we did between artists and brain researchers, but this is for another talk. We can also learn from these two fellows, Hodgkin and Huxley, because they took a very complicated phenomena at the time and tried to reduce the mathematical description of it, develop a theory of excitable membrane, and this theory amounts or reduced to 4 differential equations, 4 simple differential equations, relatively simple differential equations, but they did something that I don't want to do in my talk, and this is trying to replicate exactly the shape of this single action potential, space-clamped action potential. So they developed this model, which is a fantastic model. Really, probably the best success in computation neuroscience ever is a Hodgkin-Huxley model, because they connected the level of channels to the level of the macroscopic behavior, which is the spike. But they were trying really to imitate the action potential very, very carefully, very closely, and because they had this very stereotypic, very unified, repetitive kind of phenomena in the squid giant axon, they could do it well. They also taught us to do the following. It has to be a compact theory, 4 differential equations. It has to be biophysical theory, because I want to change parameters. So it has to be conductances that I can measure. It has to be gates that I can manipulate. It has to be biological relevant model. And it has to be predictive. As Hodgkin-Huxley, as you know well, they didn't only imitate the action potential, they also used their model to check things that they didn't put in the model, for example, a anodic break response or refractory period, which is not part of the model, but it emerged from the model, and it had a very strong predictive power to assess that the model is a very good model. So we can learn from Hodgkin-Huxley this. I want to show you something about this perfect match that is not correct for spiking neurons in the cortex. So this is our big teachers. in my case, Roll and Hodgkin and Huxley, really great physicists, great mathematicians, and also compact people, very good in kissing a la Brancusi. So some years ago, as you can see here, Bert Sackman came to Jerusalem, and he was saying exactly that. He was working then on the cortex. He was doing a lot of pair recordings in the column of the whisker system. He had a lot of fantastic work on dendrites, synaptic connectivity. Henry Markram came from there, and many other fantastic experimentalists. So they collected a huge amount of data on this cortex, huge. And he said the following: I have all this data on the cortex, cell types, firing properties, dendritic excitability, connectivity, synaptic dynamics, but I don't understand it. He really came to say, I don't know what to do with it. This is a Nobel laureate who tells you, I don't know what to do with the data. Do something with it. Model it. So for me, it was a very good way to start jumping from a single cell modeling to network modeling, working with Sackman, and later on with other people that I will mention along the way. So what we really need to do today, and that's the agenda that is emerging, is to take the structure at the fine scale level, including connectivity, synaptic connectivity. We have to know something about the connectivity, not in terms of anatomy, but in terms of functional connectivity. This synapse is doing that. This is a depressing. This is a facilitating. We heard it from Henry. We also have to speak about the firing repertoire. So each of these neurons apparently have its own type of channels, and it has its own kind of music. So it's a different instrument in a sense. And we have to understand the instruments themselves before we can understand the orchestra, the music. Without understanding that this is a violin and this is a piano and this is a cello, we can't say that we can grasp the music as a whole in terms of building the music from the instruments. So that's what I'm going to talk to you about today, if time will permit, the spiking repertoire of interneurons. So this is my team in Jerusalem. The yellow ones are working closely with Henry Markram. Others are working on other issues related to this. So my whole group now are working in this cortical column. And it's natural to go to cortical column because it's a reasonable size network, let's say 10,000 neurons. It's relatively well defined in some areas about its function. So we can start to build something reasonable, intelligent, from the single neuron level to the level of the network. So I'm not going to go today about all the work that we did about the column. I want to focus on the blue brain with Henry Markram and his team, but just to tell you that I'm working very closely with Kevin Martin in Zurich, in the Institute of Neuroinformatics there, and I'm working very closely with Bert Sackman, who now moved to Munich. Each one of us each has a different flavor about how to do this modeling work of the cortex, but eventually, no matter what people will say, we are all part of the Blue Brain project. People don't like it, especially Sackman. If I say it, he will kill me here. But he's part of the Blue Brain project. It doesn't matter the name. The essence is to build the whole circuit from scratch, bottom up, from the details to the column. No way out of it, absolutely no, because we have specific questions in mind. What do we want to understand? So we just heard a very good talk from Thomas telling us that the cat V1 cortical column and the rat V1 cortical column are behaving very differently. When you show them a sliding bar in a given orientation, in the cat, this is you with the weasel already, in the cat, most of the cells within this functional column behave rather similarly to this oriented bar. In the cat, in the rat, they are very different, and two nearby cells, very close by cells as we saw before, are saying something different. The music is different music. How could it be? The two nerve cells that are extremely close to each other, they must see something very similar in terms of environment, are filtering the environment so differently that one becomes red and another one becomes green. How can it be? How can I understand this? I can record as much as I want, and I will see all these differences. What should I do in order to ask this question? What makes a cat and a rat? And I want to understand what makes a cat and a rat. I mean V1 of rat and V1 of cat. We have to go to this level of connectivity. We have to go to this level of synaptic dynamics. We must understand the type of cells there. We must build a model of this cortical column, once of a cat, once of a rat, and look, what are the principles that allows the cat to become a cat, so that all the cells will behave similarly. Probably because of this, the cat sees better than the rat. But how this network organization? So you cannot really say that there is a generic column. There is a generic column, but sufficient variations to make a rat and a cat a cat. This is the type of question that we need this level of resolution that Henry spoke yesterday about, and this, of course, will continue when we speak about me individually versus somebody else individually, or other issues like this. We will get to this level. There is no way out of it, so I'm in it, as you can understand. Okay. Now to my specific thing about the music of neurons. So we have from Henry Markram's lab, many years in Weizmann and now in the EPFL, a lot of information about the anatomy of different interneurons in the cortical column, and also a lot of information about the firing patterns of different interneurons in the cortical columns. In this case, you can see a square pulse was given in vitro to the cells. The cells were firing like this, or like this, or like this, or like this, and people give names to these neurons. So this is a stutterer, and this is a burster, and this is a regular firing, and this is adapter, and so forth and so on. Can we objectively classify these neuron types? Are there neuron types, or is it a continuum? That's one question. The other question is, if there are neuron types, how do we automatically model these different types from the channels a la Hodgkin-Huxley to the firing pattern of this musical instrument? So this is the question I want to discuss now. One fantastic thing from Henry Markram is a very systematic thousands of cells that receive the same treatment in the beginning before of every experiment is injecting all these type of inputs into the cell. In order for the future, and here is the future, to try to characterize not only by step current of different amplitudes, but also by ramp current, noisy current, zap, chirp, noisy, color noise, different type of inputs into these neurons in order to better understand their input-output characteristics. Because, of course, in the brain, they don't see square pulse, neither do they see ramp pulse. They see something else. They see the network talking to them. So what would be the response of these cells to this different type of inputs? And how to model the cells sufficiently good so that you will have a generic model of the cell, let's say of a regular firing, that will behave correctly under all these network events. So that's the goal. So the challenge that I'm not going to talk much about today is to classify neurons and see whether there are indeed, as in the PETIA convention, which is based on the visualization of neurons, are there really different classes, or one continuum class, or two classes, and so forth? We already can see that there are several classes, maybe not 14, but several. Can we model automatically then these neurons from channels to firing? I will talk about that. Then, of course, we want to ask dynamical questions. For example, one question that bothers me a lot, and should bother you a lot, I'm sure, is why these inhibitory neurons, which are small percentage, let's say 20% of this cortical column, are so rich in terms of the repertoire of firing? Why all this channel embedded in this, specifically about these neurons? There are many thoughts. I spoke with Buzaki about that, with Giro about that, and there are some ideas, but there is no really good idea about why the richness of firing repertoire, the richness of music, is in the inhibition. Why not in the excitation, for example? And, of course, eventually you want to connect all this and ask something about the computation, how this cortical network compute gain control, contrast invariance, orientation selectivity. So I just want to tell you that at that point, I gave a talk some years ago, and there was a room full of physicists, Larry Abbott, Terry Shiznovsky, Bill Bialek, and they say here, which was amazing to me, they say, but we already understand the cortical column. These are my friends, so I can say. We already understand the cortical column. Chaim Sompolinsky. We have a model for it. So I asked them, okay, does your model explain at the same time gain control, orientation, direction selectivity, contrast invariance, because the column is doing a lot of computation at the same time. It's not doing only one thing. Of course, I can design a simple machine that does one thing, but I'm not a simple, I'm a machine, but not a simple one. I'm doing many things at the same time. So the cortical column, they don't have a good model for that, and I think we need the details in order to abstract them enough and get all these things that the cortical column is doing. So I'm in disagreement with physicists that they have a model for the cortical column. They don't. They have a model that does one thing that the cortical column can do, but not all of it. Okay, so let's go to the challenge. How to build automatically models of these different neuron types. So immediately when we start this work, looking at Henry's data, we find something that all of you know, and we spoke a lot about variability at different levels. There is a huge variability at a single cell level, even when it's disconnected from its environment by pharmacological methods. So it's an individual neuron. There is a lot of noise or variability in the system. So if you give a square pulse to this neuron in the cell body and repeat the same frozen input again and again, you get huge variability within the same neuron. Shalom Miguel. This is my brother, Miguel Nicolelis. So we cannot ignore it. It's there physiologically, experimentally. We see huge variability. I think it's connected to creativity, but we can talk about this later. I think the brain needs these sources of noise. It doesn't want to get stuck to the same behavior again and again, but that's something else. Anyway, you can see huge variability. So we cannot match trace by trace. We cannot do what Hodgkin-Huxley did and say, this is my template, I'm trying to match it. Because the same neuron will fire like this later for the same input, and the same neuron will fire like this later for the same input, within few minutes or few seconds, actually, one after the other. So how do I capture in my model the variability? I don't want to ignore variability. I want to have it in my model. So how do I do it? So it's just an example of variability. If you superimpose two traces, experimental traces, you see some spikes are missing in one, some spikes appear in the other. There is a significant variability here. So how do I capture this variability? So here is our suggestion. I hope I will be able, in the quarter of an hour I have, to highlight the principles of how to automatically capture a firing pattern, including its variability within your model, and this is the idea. First of all, we don't want anymore to look at the voltage traces, because we don't want to replicate the voltage trace. We want to replicate features of the voltage trace. And these are examples of 6 features we chose, which look very reasonable to all of us, while you look at a trace. For example, there could be these 3 first features which highlight timing aspects of this firing pattern. For example, one feature could be time for the first spike. Another feature could be rate. Another feature could be the adaptation level, so the distance between the first 2 spikes to the distance between the last spikes and so forth. So these are features that capture something about not the local properties of the spike, but the overall properties of the firing pattern, the discharge of this neuron. And you can also have local features, like the width of the spike, the height of the spike, the depth of the spike, the after hyperpolarization, and such features. So we are not trying anymore to aim at traces. We are trying to aim to capture the features that we extract from the experiment. And we define the distance error between our models. So we will have a model of this neuron. We have the experimentalist, the experimental features, let's say the rate or let's say the height, and there will be variability within each feature. Each feature has a variability. The rate is variable. The height of the action potential is also sometimes variable. The width is variable. So each feature is defined with its own experimental variability. And you define error between the model and the experiment by the difference between the model performance on each feature compared to the experimental feature divided by the standard deviation of the experiment. So the experiment will give you the range of error between the model and the experiment. And this is, I think, reasonable because you penalize the model, you assess the quality of the model according to the experimental variability. How far the variability in your model is compared to the experiment in units of experimental variability. Okay, so this is a meaningful distance. It's not just arbitrary units, it's units of the experimental variability. So that's the idea, and we call it the mu system. You should remember mu from my talk. Cannot go from Sweden without mu, not because there is amazing cows, but because the mu system is the multiple objective optimization approach. And here is the idea. You use features of spiking, several features, simultaneously, let's say 6 features of the spiking pattern. You assume channels, ion channels, that may exist in your neuron. Okay, so we have now some literature that is rather solid about the type of INA channels, K channels, calcium channels, M-type, H-type of channels. You put them in your model as a parameter. You assume that you can change only the maximal conductance of each channel. So we don't play with kinetics, although of course, this also could be a parameter. But for this simulation I will show you, I'm going to only to play with the maximal conductance of each channel. And my genetic algorithm will search for the correct conductance to give you the minimal error between the model performance and the experiments. And then we are using to do this multiple optimization system or approach, because we want simultaneously to satisfy both the rate and the height and the time to the first spike. We want to simultaneously satisfy 6 objects in this case. So it's multi-object optimization. Okay, we are looking for a method that will simultaneously find the balance or the trade-offs between different objects in order to minimize the error in all of them relative to the experimental variability. Okay, and then we use several genetic algorithms, simulated annealing, and other automatic ways to reduce, to find the best model, to converge, to constrain the model until it hits the best it can into the experimental variability. So this is the system. The mu system actually, let's say that we have, let's say, two objectives. Let's say the strength of the steel and the weight of the steel, if you're an engineer, or let's say the density of one channel, or let's say one feature will be, let's say, the height of the action potential, another feature will be the rate of the action potential, and you want to satisfy both of them, or actually 6 of them in this case, and you are looking for what combination of ion channels will give you the best possible trade-offs, and this is what is called the Pareto Front. So the Pareto Front here is the best trade-offs that you can get for these 2 objectives, and then you have to choose, you know, do you want less mistake, less error in this objective, and more error in this objective? Do you want to be here and give them equal importance? Then you will get this error in this objective and this error in this objective. But the optimal solutions will lie here, and then it's your preference whether you want the width of the spike to be exact and the rate to be less exact, or do you want exactly the rate and the width of the spike less exact. This is your choice. But at least the model gives you where you should look at. And this is a personal preference because you cannot satisfy all of them, of course. You can satisfy some of them on the account of others because there is a trade-off between these objectives. And this is how it looks. I'm not going to go all these techniques, but for example, if you want to satisfy in some type of neurons the action potential overshoot or undershoot or after hyperpolarization and the accommodation, you will get this Pareto Front. And in this case, you see that this point is optimal for both. So if you have a Pareto Front that looks like this, then it means that there is a point, that there is no trade-off. You can satisfy both the best way you can. In some other cases, for example, the spike rate and the accommodation index will give you a Pareto Front like this. Then you choose whether you want a better spike rate, closer to less error of the spike error rate versus to experiment, and more error in the accommodation index, or vice versa. So this is what the model can do. It can do it automatically. You just give it from the experiment a set of features. The model will run for you with these channels and then give you which kind of combination of channels are falling within this Pareto Front. Okay? So let's take a case study. We took actually 2 neurons that are very clearly also in our classifying algorithm that there are actually at least 2 types of neurons. They're accommodating and the fast spiking. We took them 2, and we tried to do this running of this mu. And first of all, to our amazement, I must say, as I said, we did not try to fit transients. We are not trying to fit voltage. We are just trying to fit features of the voltage traces. So this is the experimental one, which the brown traces. The green traces is one manifestation of the model. Of course, there are many manifestations of the model depending on what points in the 6-dimensional space you choose as an example. Just one example, and you can see the traces. They are really, really looking very close. Experimentalists cannot tell you whether the green and, I mean, beside the little noise that you have in the experiment, the green and the brown are coming from 2 same experiment twice or from a model and an experiment. So the whole trace is now rather well, not exactly, and you don't want exactly matching, but the whole trace is matched well just because we chose 6 reasonable features to match. So the model of these 2 cells are doing very good. Just to give you an example, not only very good, but it tells you, for example, for the accommodating cell, you get in this kind of 3-dimensional, this is a sodium conductance, this is a potassium conductance, this is another persistent sodium conductance, and you can see that for some parameter space, within this 12 or whatever number of channels, some die out because they're irrelevant for the behavior, some are relevant to the behavior, but you can see that these 2 types of cells, 2 types of firing patterns are separated automatically by this method, at least in this 3-dimensional space, and you can see that there are many good solutions, many combinations of channels that are acceptable if you assume some kind of standard deviation error that you agree that is acceptable. So all these solutions for square pulses are okay for one type of neurons. All these solutions are okay for the other type of neurons, and we could be satisfied with that because they are producing nice traces. Hodgkin-Huxley will say it's a good match. It's not spike by spike, but as I said, we don't want spike by spike. Okay, I just want to highlight the fact that Eve Mader, this fantastic work, using messenger RNA showed that for the same type of neuron in her system, which is not cortical system, but for her type of neuron, the biology also solved the firing pattern, one firing pattern with different combination of ion channels. So biology does not have one solution for a given firing pattern. Different combination of channels in biology, different combination of channels in our methods will give you the same firing pattern. But as you can see, they are much more constrained here in terms of space relative to here. But this is because we didn't constrain our model good enough, and we know how to do it better. So is there an optimal stimuli, rather than the step current pulse that we were using until now, to constrain your channel density model better so it will satisfy many other inputs that you did not use before? So from Henry Markham's lab, we have a lot, as I said before, a lot of inputs. For example, many square pulses with all its variability, many ramp pulses with all their variability, but of different amplitudes. So we have a range of inputs, and we want to satisfy many simultaneously, not only for the square pulse, but the firing pattern also for the same neuron getting another input. And I want to show you very fast our recent results, and this is the following. So let's start to train our model only with square pulses. You give one square pulse, okay, you get, this is the training session. You train the model on one square pulse in order to get the best match between model and experiment. This is this point. Now you want to satisfy the model on 2 square pulses, the first and the second square pulse. This will give you a higher error because it's hard now to satisfy both the firing for a small pulse and also the firing for a higher pulse. So your training error increases always, and this is any machine learning people know that when you have more targets to satisfy your training, typically your training error goes up. But here at this point, our training on the 4 inputs are good enough for the 4 of them simultaneously, for the output of the 4 of them simultaneously. Now you want to generalize to an input that you didn't see before. So if you have only one training session on a small input, you don't succeed to generalize well for this bigger input. But if you train your method, if the mu is being trained on 4 inputs, the training session is a little bit worsened, but the generalization becomes better. So after training of the 4 of them, you get a very good generalization, a very good prediction on how your model and the experiments will behave on this input. So that's what Hodgkin actually did. They trained, so to speak, their model on the shape of the action potential, and they want to generalize and see if it succeeds to give them, let's say, anodic break response or refractoriness. That's exactly what we do here. There is a training session and then generalization session. The fact that generalization becomes better means that the training is improving your generalization power. We do the same on ramps. We train the system on 4 ramps, output of 4 ramps. You get worse as you have more constraints. But then you want to generalize on another ramp. You get an improvement. Okay? Now the question is, if I train only on steps, can I generalize on ramps? So I'm training only on ramps, and I'm asking, can my model generalize on an input that it never saw before? This is called generalization from one input type to another input type. And that's what we are doing. So we train here. This is the experimental. We train. We get the model response, which is good for all these kind of inputs. This is one example, one manifestation of the model. Then you try to predict the behavior of the model to a ramp pulse, and you want to compare the model behavior to an actual experimental output for this specific ramp input. Okay? So you train on steps. As I said before, the training becomes a bit worse as you train on more and more steps. But the generalization on ramp is very good. So if you train on ramp on step, you get good generalization on ramps. And this is an example. So this is training on steps, and then you give a ramp input to your neuron, and the model really fits nicely, again, with the variability that you find experimentally, to another input. Surprisingly, if you train on ramps, and you get this worsening in the training, you don't succeed to get good generalization on steps. So it's not good to start training on ramps, because you can get good fit only for the ramps, but when you try to generalize the model on steps, it's not good. So from ramp to step is bad. From step to ramp is good. So it's not symmetrical, this kind of machine learning type of approach, and that's why we need to have what kind of combination of inputs will give us the optimal way of training so that we can generalize to any input that you want later on. And this is what we are doing here. So we train only on ramps, only first ramp, then we train on the first and second ramp. Of course, we get better constraining of the channel types. Then we train on one step, second step, alone, and then we want to satisfy all of them together, and that's what happens. You get a constrained region. Now the channel, the acceptable solutions become narrower and narrower. As you get more and more type of inputs to constrain, type of inputs to satisfy, you will get a smaller range of ion channels that will satisfy all these inputs. So let's take point from here, which is now a superposition of the good solution for only ramps and for only steps. This is good solution for both of them together. Let's see how we generalize to noisy input and network input in our brain. So this is the input we are using. This is a color noise. Now we given the same neuron that saw the ramp and saw the step and was trained on both of them. Now the model should satisfy the response of the same neuron experimentally to the same input theoretically. So this is the red one is the model response. The blue one is the experimental response. We can now generalize very, very well, really, really well, within the error, experimental error, to inputs that seems to be more relevant to what the neuron sees in my brain when I talk now to you. So this is the input that my cell is receiving. This is the experimental, the blue lines, the blue raster dots are the output of 10 repetition of the same frozen input experimentally, this is the response of our now constrained model by both step and ramp current. You can see this is one manifestation. Of course, I could show you many. And now you have ways to compare it. And you can use Shiznovsky and other methods and see that actually your model now predicts extremely well, 90% reliability, as they call it, between the model variability and the model variability and the experimental variability. And interestingly, if you use an integrate and fire model, a simple integrate and fire model as your model and ask yourself, okay, so I give now this input to my integrate and fire model, how good the integrate and fire model is doing compared to the experimental data, it's doing very, very bad. We cannot use integrate and fire model, at least not these simple ones, in order to replicate what the conductance-based model is doing. So I want to finish by summarizing, and later, if you have time, I'll tell you what I think is the agenda. I want to say that these conductance-based models lend themselves to this automatization of constraining model parameters. We have to take into account experimental variability, so we don't want to match trace by trace. Okay, MOO is a very good method. You even call it MOON if you want, because it's multiple objective optimization neuron methods. You can do it automatically, very fast. Within a few minutes, we'll get a range of models that will satisfy your behavior. Generalization is very important, as I said before. So training is very important. You don't want really to match what you put in. You want now to match something you never saw before. What I'm doing now with my colleagues and with Henry is to do both. Still, we are not completely happy with the combination of first step and then ramp. We want to find a better input so that all experimental team in the world will use this input and give us the output, and we shall automatically tell them what are the conductance base that produce this thing. We are looking for this optimal input now, and I think we are getting close to it. And also we want to objectively classify neuron types using features. So we shall use the same feature idea, and it can be extended to more than 6 features in order to see whether we can cluster and get different clusters rather than a continuum using some kind of features, rate, and other aspects of these firing patterns. So features will be also used to classify neuron types, not only to model a specific neuron type. Okay, so I know that Eric wants me to go, but you don't want to eat, I'm sure. So I just want to say one word, and by this I end. I really think that artists are better than us in terms of changing the world. Really. You can see, for example, the Dada movement that came at a given period of time. They called themselves Dada. It's not exactly clear why. It's an unclear name. It could be nonsense word. It can be yes, yes in Romanian. It can be hobby in French. It doesn't matter. But as Marcel Janco, who was the head of the Dada, said, there are times to change the culture of how things are happening. To change the culture. I think we, as scientists, are extremely conservative. We are actually relatively happy with what we have, and we should not be happy with what we have. First of all, we did not succeed to collect data correctly in the last 100 years. And this is not it. You can do as much databases as you want. We have to decide to put our data there. Why should I put my data there? Because I need to get a professorship. Why should I put my data there? It's my data. It's my neuron. It's our fault that we did not do it, not anybody else. So we should share data like the Dada shared a manifesto. We should do it. And we did not do it in the last 100 years, and the Blue Brain Project is one way to do it. But seriously, not in talking, in doing. That's one aspect. The other aspect is that most of us do not believe that we will understand the brain. So we are doing little things, and we are happy with what we do, and we publish in Nature. But this is not good enough. We have to start to work in groups. We have to have movements. These are also very individualistic people, no less than us. But they did work in groups for a period of time. They solved some problem. They have a manifesto. They work together. They created a movement. They changed the world in their period. Then they moved to something else. We should do it now. So I suggest to establish the BABA movement, Brain and Behavior Agenda. Not Dada, but BABA. Thank you very much.
So we have time for maybe one question.
Okay, so I'd like to take a position because I can always do that very well with Idan. And the good thing about Idan's talks is that it introduces all sorts of objections in me. So Idan, so I think that you lead us in the wrong direction by taking a machine image.
By taking, sorry?
By taking a machine image. I mean, you point to a new neuroscience, and then you use an image that comes from the beginning of the industrial age. And the reason why I think that the machine image is bad is because it presupposes a certain kind of causality between the elements, which we see is really not a way of understanding the biological mechanisms of life as we begin to understand them. It's not so causally clear. Right? And so you take for your image, for example, the orchestra of neurons, of defined components producing the music as a result, which is, once again, quite a strong machine image, whereas we know, and you yourself are involved, in looking at the encodings produced by those instruments, and we know that the music is actually constraining and changing the underlying form of the instruments. This is what we expect, which is really not a typical machine-like process. So I wonder whether you're choosing the right image here, and I would like to have this debate. This might not be the right time, but...
Okay, so we can discuss this, and first of all, I'll be happy to get another metaphor from you, if you have one, rather than a machine. The heart is also evolving, the muscles are also evolving and changing, and the music of the brain is also changing while I'm talking to you, and after not sleeping or sleeping, it will be a little bit different music. The fact that Miguel Nicolelis can move the robotic arm of the monkey with changing music means that he also realized how the music changes during plasticity. So it's still a music of an orchestra that is training, and while training, without a conductor, but on its own during behavior, still it produces a music, this system. The music is changing. There are rules for the change. This rule should be implemented in my machine, and it will be a music machine with improved music to a certain degree.
Yeah, fine. So there is a way that you can argue this, right? But,
I'm sure.
Yeah, but there's some serious problems we have to face here. For example, the generation of presumably a physically bounded mind of qualia, right, of subjective experience, something we have no understanding of in conventional machine technology. So all I'm arguing is that let's be careful, let's try and move the image in the correct direction. And
I think the qualia is our invention. It's an invention of the machine. When we shall understand the machine, we won't need the word qualia anymore. It will emerge from the machine. By the way, I'm not saying that I can predict the machine that I will build. It will also have what you call qualia.
But that's
I also call qualia because it's from the ignorance of what does it mean qualia. Okay. It's nothing beyond this machine.
It is very exciting, but we actually have a poster session that's supposed to have started 10 minutes ago.
Thank you very much.
So
anyway.
in the interest of... So let's applaud for Idan again. And let's get lunch and the poster session started.